So, as Dr. Sherman said, today I'm gonna tell you about computational approaches that are part of the bigger framework of system biology that we have used at the Baliga lab to study the pathogen with the idea of trying to find promising drug targets, understand the biology, what are the drivers of some of the strategies that allow the bacteria to thrive in the host environment. In that way, we can use that knowledge to design new therapeutics, and in that way, tackle the TB pandemic. So I'm gonna be presenting today the different projects that have been developed in the Baliga lab, and there is a lot of people involved that you can see some of them here, and we have a lot of collaborators too. So this is a part of a big group of people working together on this. And we have covered this, but just as a reminder, TB is a big burden, being the leading cause of death by an infection disease. And importantly, there is a significant overlap with another public health crisis, that is the antimicrobial resistance. In fact, TB is about 25% of the cases of antimicrobial resistant death. And for the multi-drug resistant TB, only about 50% are cured with the standard treatments, and those treatments can be really long. They can take from 6 to 30 months, and that's a huge burden on the patients that has to take a significant amount of pills. And we already heard about side effects that they may have. Going back to the antimicrobial resistance, just to illustrate the importance of tackling this issue right now, these are some estimates of what would happen in 2050, and it has been estimated about 10 million annual deaths due to AMR. To give you a context of how much is that, that's more than what was estimated at 2016 when the study was commissioned, of people that die due to cancer. So that's a big concern. And as I said before, TB accounts for about 1/4 of the current AMR fatalities, that only in 1 year for 2019 was already estimated, depending how you counted, more than 1 million or even up to 5 million people dying due to AMR infection. So this is a big problem, and was part of the motivation. So initially at the Baliga lab, the starting point was this question of why treating TB is so challenging, and how can we find a path forward, taking into account the heterogeneity that we see in the bacteria. And just to illustrate the point, this is a cartoon for a solid granuloma, where the bacteria is considered to be encapsulated and surrounded by different host cell types. And importantly, this is not an homogeneous population. You may have subpopulation within this structure. And what we think that is relevant is that you may have a really good drug that may target a large part of the population, but may not target all of the population. For example, this drug one is pretty good at killing subpopulation in orange color, but if you are not able to target all the subpopulation, let's say the red ones, some of them die, in this case the red one, but then you have others that survive, those are gonna continue the infection. So we really want to answer these two questions. First is, how can we rationally formulate regimen of drugs that are individually targeting different subpopulations, that when combined targets the bacteria population as a whole? And also, can we find a better way to estimate the efficacy of treatment? With the standard methods are culture-based approaches that are really time-consuming. So the time that goes from taking those samples to really design a patient treatment is really too long. So if we were able to find a quicker way to say if a person is responding properly to a treatment, that will allow the patient and the clinician to adjust the plan in a faster way, hopefully improving outcome. So to do that, as we said before, we are using computational approaches. So one big part of our effort was transcriptional networks, and we have applied this to different bacterial species. Specifically, we developed a EGRI model, that is EGRI stands for Environment and Gene Regulatory Influence Network. And with these models, we want to do two things. We want to recapitulate what we know and what we are measuring and putting into the model, but we also want to have a predictive power. We want to know what's going to happen in new contexts that we have not profiled yet. And that is especially important in the context of NTB, where we may not be able to profile every host, every infection relevant context. And once we have this transcriptional network, and this is just like a map of how the gene expression is controlled. So if we take all the genes encoded in the NTB genome, now we can identify what are the transcriptional regulators of each gene and what is the transcription and what the expression profile of these regulated genes among different conditions. So in that way, we can identify promising drug targets, because then we can see these genes are really responding to a particular condition that may be infection relevant. So if we have that information, we can move forward. And I will tell you a little more with an example. Also, by combining the transcriptional network with a second layer of information as the metabolic network, now we can really see what is the implication of perturbing genes or transcription factors that are the ones that control the gene expression at the RNA level into fitness. So in that way, we can see which TF, the transcription factors, are the most promising targets for perturbation. So in order to do that and really be able to profile host infection relevant contexts, we have also developed different technologies. Vivek, a former postdoc in the lab, he developed the PERSORT technology, where he was able to isolate and study this pre-existing persister. Basically, he identified this subpopulation that is transcriptionally active but translationally dormant. And what he showed is that these persisters are highly tolerant to antibiotics, and there are some transcriptional signatures for this subpopulation. Similarly, we have developed some reactors, and in this one in particular, we were interested in hypoxia, another condition relevant for the infection context, especially in the environment of the granuloma, and we were able to really have a high resolution of what are the different transcriptional states that the bacteria may adopt when they are during entry and exit out of hypoxia-induced dormancy. And Eliza Peterson also developed the PAPSI technology that allow us to enrich for the bacterial transcripts from samples of infected cells. In that way, we can really know what the bacteria is doing within the host. So that is all information quite valuable for developing the models that we develop. And just to illustrate a little more what we can get, what kind of information you can get from this type of technologies with the bioreactor, what we did was we started with a high level of oxygen. We decreased that. And then once it was 0, so completely hypoxic, we stayed there for about 2 days, and then we start putting oxygen back. And what we identified is that there were really 6 different transcriptional states adopted by the bacteria during that process. There were what we call normoxia, the high oxygen concentration, depletion when the oxygen was going down, 3 different states of hypoxia, early, mid, and late hypoxia, and then what we call resuscitation when oxygen is entering again into the system. And what we found is that we could identify a specific transcription factors that are relevant for the transitions between these states and to sustain these states. And also we saw specific architectures in the network that seems to be key for the transition between states. Also, we can get some biological insight. For example, here for what we call the late hypoxia that has about 978 genes. This is the average profile of those genes. So at the end of the hypoxia, we see this upregulation of these genes goes down, and we identify an enrichment for these mammalian cell entry related genes. So when we were asking why would that make sense, we hypothesized that this could be a way for the bacteria to prepare to exit out of the dormancy and then be ready to infect new host cells. Okay, and as I mentioned before, we want to develop these predictive models. And the approach for that is we need to do perturbation in the system. We need to observe and measure what those perturbations lead to, and then we can model those. So for example, here, and this was the first network developed by us for NTB, we can have different types of inputs like the pH, oxygen, different drugs, and we can see what is the phenotype. And we also can then, by taking all these genome-wide transcriptome, develop a network, a transcriptional network. With that transcriptional network, we can identify groups of genes that seems to be co-regulated. And then we can importantly model which are the regulators of those genes. And we can make new prediction about what is gonna be the expression profile of those genes in a new condition. And that could be based on the expression of a given transcription factor, or could be based on a variable, for example, depending on the oxygen level. So when we have that, we can make prediction, and then we can go do the experiment and compare what we predicted with what we measure. And with this approach, we have shown that we can have a high correlation between our prediction and the measurements that we do. And as I said before, we can get here a layout of the network that allow us to interact, how the different components are coming together and interacting between them. As I said, once we have those, we can do new analysis. And here, as I said before, another component is now connecting and integrating the transcriptional with the metabolic network. We did that with Prime. Prime was developed by Rupa. And basically what we can do is we can predict growth rates. We can also predict in vivo, intracellularly, what could be the growth rate in different types of cells, for example, alveolar macrophages versus bone marrow derived macrophages. And we can also, as I said here, check what happens if you knock out each of the transcription factors or genes in the genome. And then we can see which ones are the promising targets, and then we compare this to actual TNC data that measure what is the fitness consequence of these gene disruptions, and we see a high correlation. And as a second example, Eliza Peterson recently published another paper where she found this set of genes. Also, this is another visualization of an E-green network. And what she found was that this set of genes have these gene regulatory elements, and among those, she saw that this one was highly similar to a motif reported for this transcription factor, NTRA. What she saw is that when she knocked down NTRA, there was this change in the morphology of the bacteria, the length increases, and also importantly, by knocking down NTRA, you see a much higher susceptibility to antibiotics. And we saw that for both isoniazid and rifampicin here. So in that way, we are finding new drug targets that we can use to identify then new molecules. So once we have these promising drug targets, we can go and try to identify molecules that work in those targets, or identify new molecules or even rationally design those molecules so that we can, put forward potential new compounds. And just to show you what is the general framework that we have and how we think about this, we have here our susceptible population that is treated with the primary drugs. But this is not a single state. Actually, these cells can transition into other states, as I said, the persisters, or they can move to the resistant state. And what we want to do is we want to either prevent those transitions from susceptible to resistant and persisters, or reverse that. We have shown in other species that we can actually target compensatory mechanisms of resistance, so that the fitness cost is so high that this anti-resistant phenotype is not sustainable, so they are gonna revert into the susceptible. Quickly, I just want to tell you a little about the second part, that is now how can we evaluate efficacy during a treatment. And as I said, we want to move away from culturing And Vivek, a former postdoc in the lab, developed a transcriptional signature-based approach that could compute a cell viability score. So you just put a transcriptome and it will tell you what is the viability of that population based on the transcriptome. And what he showed is that compared to the most probable number, the cell viability score was better to evaluate if a population was actually viable compared to the CFU count. CFU was misleading here, suggesting that you have a significant decrease in the bacterial population where the MPN approach show is almost the same. But and we see that cell viability score was able to capture that. And he also showed that but using the cell viability score, you can identify which patients after 2 weeks are responding to treatment and they have a decrease in viability score versus the ones that are not responding and actually have an increase in the viability score. And now that we have this cell viability score, we can, for different drugs, estimate this. And Vivek also wanted to see, can we then do an in silico prediction of what would be the cell viability score so that we can identify the most synergistic combinations. So that's what he did then. Once he had the DRONA, he developed basically a cell viability score for each of the different compounds that he wanted to combine. And then he did an in silico combination. And in that, you get a combination score. You can identify, we can predict which combinations are synergistic, which ones are antagonistic. And he showed with experimental data that indeed there is a significant difference in the score between synergistic and antagonistic combination for 2 drug combinations and also for 3 drug combinations. And this was in collaboration with Bree Aldrich. So with that, I just want to summarize that I show you today a few examples of multi-scale system approach that we have been using to dissect the drivers of the adaptive strategies that the bacteria may take within the host. And we do that by profiling different transcriptomes, different host relevant contents, inferring a network, then trying to identify the regulatory mechanisms acting on those phenotypes, and then we can predict consequence of what would happen when you perturb that. Using prime that integrate transcriptional and metabolic networks, then we can also identify the mode of action of different drugs. We can do prediction about what will be the effect of drug combination, and we can also rank order targets for potentiated drug treatment based on the consequence that they would have in the fitness. And the second part, I show you a few examples of machine learning-based approach and how we can use that to track treatment efficacy and also reduce the burden and the resource needed to at least narrow down a set of potential synergistic drug combinations using DRONA that define the cell viability score and ML Synergy that then combine in silico these different scores from different compounds to evaluate which one would be synergistic. And with that, I just want to thank everyone in the Baliga Lab, current and previous members, all our collaborators, and our funding. That's all. Thank you.
Thank you.